Hello, we're about to begin our video. In this video, we'll take a look at how carbon contributes to the diversity of life on the planet, because it's the backbone of molecules in living things. So we're going to take a look at molecules that have carbon as their skeleton and how they can vary. And there is a figure that's worth looking at. So I'm going to slip over to that figure that describes the things on this slide, or that's described by the words on this slide. Okay, so in this picture, we're looking at molecules that are all hydrocarbons. Hydrocarbons are molecules made up of carbon and hydrogens, and that's it. But they can vary in length and in position of double bonds. Let's take a look. And as they vary, they're probably going to be different behaviors of the molecules. So we can look at a molecule that is 2 carbons long or 3 carbons long. You can make a molecule 5 carbons long or 6 carbons long. In part B here, a molecule could be straight or it could have a branching pattern to it. Butane and isobutane have the same number of carbons and the same number of hydrogens, but they have a different shape, don't they? Because of this branch that comes off of carbon 2 here, all right? Do you think that changing the length or the branching pattern affects the behavior of these molecules? That's kind of a weird word to use with respect to molecules. Behavior. Can molecules behave? Well, sort of. If you think back to what you do in chemistry classes, you might look at how molecules behave differently by measuring things like their boiling point or their melting point, or their mass, or their freezing point. Changing the length of the molecule or its pattern, its branching pattern, will likely change the way that this molecule behaves, increasing diversity in molecules. In the part C here, you can see that sometimes carbons have a double bond between them, right? That would be two pairs of electrons that are shared between these 2 carbons. And that position of the double bond can vary from one molecule to another. So here we've got 1 butene and 2 butene, and sure enough, they have the same molecular formula, the same number of carbons and hydrogens, but they have a different structure. And probably because they have a different structure, they're going to have a different behavior, contributing to diversity. We can even take a linear molecule, and sometimes it forms a ring, as in cyclohexene here has a ring shape, or benzene has a ring shape. And this reminds me, when you're looking at molecules, sometimes you just see this kind of bare backbone, just lines. And what does this tell you? Well, it tells you that at every intersection is a carbon. And each carbon forms 4 covalent bonds, so that this carbon here, at this intersection, must have a bond to another carbon, 2 bonds to another carbon, I should say, and then it must have 2 hydrogens attached to it as well, and sure enough it does. This is kind of the shortcut way that we write molecules when we're kind of lazy. Okay, so these molecules, by varying the length or the position of a double bond or the bond order, you can have different kinds of molecules. They probably have different functions or different behaviors. We can also increase diversity by thinking about isomers. A couple of the molecules on the last slide were isomers, but isomers allow carbon molecules to be even more diverse. What is an isomer? Well, just what is an isomer? The word itself, isomer, mer means parts and iso means same. So this is actually saying, same parts. Isomers are pairs of molecules that have the same parts. And there are many different kinds of isomers. We'll look at just a couple of them. We're going to see some in later chapters as well. One of these are structural isomers. Structural isomers are isomers, a pair of molecules, that differ in their bond order. So they have the same molecular formula, they have the same atoms, but they differ in their bond order. Different atoms are bonded together. Let's take a look at an example. Two molecules here that we're going to see in Chapter 5 are glucose and fructose. Glucose and fructose both have the molecular formula C6H12O6. Okay? But they're different because they are structural isomers of each other. Notice the position of this C double bonded O. Okay? In glucose, it's on the end. That C double bonded O is called a carbonyl group. In fructose, it's more in the middle. It's not on the first carbon there. It's in the middle. And that changes the behavior. The molecules glucose and fructose are treated differently inside our cells. They are both simple sugars. They both can provide energy, but they're treated a little differently because they have a different structure. So changing the bond order, making structural isomers, can change the behavior of the molecule. That's one type of isomer. A second type of isomer are called cis-trans isomers. Isomers that have the same covalent relationships. So you can put in here if you want to, covalent relationships, but differ in their spatial relationships. So we've got the same bond order, but we still get two different kinds of molecules. And in the molecules we're going to study, we typically see that the atoms assume a different position around a carbon-carbon double bond. So you've got a molecule with a carbon-carbon double bond in it, and the atoms assume a different position around that rigid double bond. Two carbons with a double bond between them, they aren't flexible. Those are pretty rigid. And a cis-trans isomers go something like this. The cis version of the isomer has our two functional groups, if you will, on the same side of the rigid double bond, of the carbon double bond. The trans isomer has these two functional groups on opposite sides of that double bond. In this picture, we're not looking at a real molecule. The X's represent some functional group, so it's kind of weird looking. But what this does, the cis and the trans isomers act differently inside cells, inside living things, or they can act differently, making diversity. I wanted to give you an example of several molecules that are cis-trans isomers and how they function differently. And it is an example that leads into Chapter 5 when we start talking about fats. Fats are a type of lipid, and there are different kinds of fats out there. You've probably heard of saturated fats, unsaturated fats, and trans fats. Saturated fats have fatty acid tails, and these tails are long chains of carbons with hydrogens at every single carbon. They're saturated with hydrogens. We find saturated fatty acids in animal products like butter and such. Okay, but we're not really going to focus on that right now. It's the unsaturated fats that are kind of interesting. Unsaturated fats have at least one carbon-carbon double bond within this long chain of hydrocarbons. This one happens to have two, actually, two carbon-carbon double bonds. And so we say that the carbons are not fully saturated with hydrogens. They're unsaturated because these two carbons, for example, have a double bond rather than being bonded to another hydrogen. Well, what's interesting about unsaturated fats is they can exist in two forms, cis or trans. In nature, Most of the unsaturated fats out there are cis unsaturated fats, causing kind of a bend or a kink in the molecules. We find unsaturated fats in plant products. So the oils like olive oil, canola oil, vegetable oils are composed in large part of unsaturated fats. And our body does well with them. We need unsaturated fats. We use them effectively and efficiently. Now, in processing of unsaturated fats, humans have made what are called trans fats. Trans fats are mostly human-produced fats that are kind of like unsaturated fats and kind of like saturated fats. They are unsaturated fats in the sense that they have a double bond between two carbons, but that double bond is in the trans form, creating a much more linear molecule, much more like the saturated fat, as you can see. Okay? Trans fatty acids aren't as good for us. When we eat them, our body doesn't know exactly how to deal with them as well. And so they are much more like saturated fats that we eat. Yeah, it's okay to have some, but we shouldn't be loading our diet with trans fats or saturated fats because these two molecules act differently in our bodies. Cis and trans unsaturated fatty acids act differently. Okay. One more type of isomer before we move on. That's enantiomers. Enantiomers are isomers that differ in, let's just say isomers that are mirror images of each other. So we have molecules that have the same bond order and the same covalent relationships, but they're mirror images of each other. So there's like a left-handed and a right-handed version of each molecule in this pair of enantiomers. And we've got a picture here of a pair of enantiomers. We call one the left-handed isomer, the L isomer, and one the right-handed or dextro isomer, the D isomer. And these molecules happen to have a central carbon surrounded by 4 groups: an amino group, a methyl group, a carboxylic acid group, and this one happens to have a hydrogen as well. Okay, but they're not exactly the same because one is a mirror image of the other. There's a left-handed and a right-handed version. These particular molecules are amino acids. This is one particular type of amino acid, the left-hand version and the right-hand version. Amino acids have an amino group and a carboxyl group on a central carbon. And this particular amino acid is, I believe, alanine. Okay, so do you think that these molecules behave differently, like this left-handed version versus the right-handed version? I mean, they look exactly the same practically, right? They're just mirror images of each other. Well, it seems like they do behave differently. Inside us, we use amino acids to build proteins. There are 20 different kinds of amino acids, and we use them to build our bigger molecules called proteins. Guess what? Our bodies can only use the left-handed version of each amino acid to build its proteins. And so the right-handed version of the amino acids are not used. Kind of funky. Here's another example of how structure relates to function with respect to enantiomers. So maybe some of you have heard of Parkinson's disease. People with Parkinson's disease have a lot of very specific characteristics, right? Here's a little man who has rigidity, and his body is not, he kind of shuffles when he walks. He might not be able to show many expressions on his face, and he might have some shaking as well. What's really going on is there's a problem inside his brain. It's a disease of the basal ganglia of the brain, and that part of the brain is involved in the control of movement and also cognition. And there's an important neurotransmitter in that part of the brain called dopamine. What's happening, we think, with patients, Parkinson's patients, is the cells that make dopamine are dying. So they're making less and less dopamine. And that is why some of these motor-related symptoms show up. So how do we solve this problem? It'd be nice if we could just give Parkinson's patients dopamine. Here, have some extra dopamine. That will help you reduce the effects of your dying brain cells. But can we treat people by giving them dopamine? No, we can't. Dopamine is a big molecule, and it can't breach, or it can't pass through the blood-brain barrier. And so we can't give people dopamine. But instead, we can treat them with a precursor of dopamine that can be converted into dopamine in the brain. That's much smaller. L-DOPA is that precursor. We can't, it turns out L-DOPA is a molecule that has an enantiomer. We can't use D-DOPA for this treatment. We can only use L-DOPA. Only L-DOPA can be converted into dopamine in the brain, and it does help, right? So why must patients be treated with L-DOPA? Because the D-DOPA version of this enantiomer can't be converted to dopamine. They're different structurally, and so functionally these two molecules are too. Does the treatment work? It helps, absolutely. It slows those symptoms down. Does it cure Parkinson's? No. Okay, brain cells are still dying. Dopamine is lost with all those brain cells that are dying. There are some new treatment options. One of them involves transplanting new cells into the brains of Parkinson's patient, cells that can develop into stem cells, that can develop into neurons that make their own dopamine. That's pretty cool. Okay, that's just one example of how having two different isomers have very different effects inside living cells. We'll have one more video coming up.